and extension entomologist with Iowa State University. I'm also the moderator today. And along with Dr. Justin McMecken, he's an associate professor and extension entomologist at the University of Lincoln, Nebraska. And Dr. Bob Cook, he's a professor and extension entomologist at the University of Minnesota. Just wanted to give out some welcomes, introductions, and thank yous before we get started. Of course, none of this research and extension would be possible without a number of supporters, specifically the North Central Soybean Research Program, and then our state soybean boards, Iowa, Nebraska, and Minnesota provide a lot of support for us to do research and extension. We have piecemealed a lot of federal grants to support student training, like the USDA NEFA, Crop Protection and Pest Management Program, the North Central Region Sustainable Agricultural Research and Extension, and then the North Central IPM centers. And many of you probably work for industry, and we just really appreciate the chances to get seed and chemical donations and also some financial. Some of you out there may have allowed us to sample or do more extensive research on your fields or your farms, and we really appreciate that. We appreciate it. It's a risk to your soybean field to let us basically let midges thrive, but it helps us learn and find out some of the possible management strategies that might be working for this pest. So we really appreciate you for letting us use your farms. And then we would also like to acknowledge Phyllis Bongard. She's from the University of Minnesota Extension. She's in the background today. Been doing this for 5 years. Our format today is going to be similar to what we did last year, where we prepared a series of short talks to give you sort of the latest and greatest research updates with frequent breaks for questions. And our first speaker today is Dr. Justin McMecken, and he is going to be talking about questions that almost everybody has when we talk about soybean gall midge is what's the current distribution and monitoring for this pest? So thanks, Justin. Take it away.
Okay, I'll start us off today just with a brief kind of covering a couple things, distribution and monitoring, but there were several questions that came in as many of you registered about kind of key topics that are a little bit of background, but are always kind of changing on soybean gall midge. And so a couple slides here to answer some of those questions. And hopefully those of you that have seen or know soybean gall midge for a while, a bit of a refresher on that side.
Yes.
Okay. All right. So this slide will be familiar to many of you. It's available on the soybeangallmidge.org website. There's some updates here. If you haven't been keeping track of this year, we did identify 14 new counties in every state of the major states where we have issues. So Nebraska, Iowa, Minnesota, and South Dakota, and then Kansas as well. This whole area encompasses about 19.3 million acres. And when I say that much area, if we find a single larvae in a county, we highlight the whole county. So this is definitely a high estimate in terms of what we see. And then in Nebraska, as well as Minnesota, hiding behind that percent sign is some dashed colonies you'll notice here in 2024 or from the 2024 surveys. And that dash line means that we found it on sweet clover, but not on soybeans. So the other hosts associated with this insect are important to pay attention to when we're looking in the field. And there's great webinars from the past for you all to look at if you want to see more about those hosts. The other thing I've highlighted is just the percentage of acres at the county level in that map that now are considered to have soybean gall midge. So 90% of Nebraska's acres, 76% of South Dakota, 75% of Minnesota, 61% of Iowa, and then single digit numbers for the other states that are out there. If you're not familiar with soybean gall midge, maybe you're on the outer perimeter of this area, just know that not all these areas are experiencing significant amounts of pressure. That's actually quite a bit lower in more concentrated counties. I'll show you a little bit of survey done in Nebraska through the Nebraska Soybean Board. But we do find the insect, at least in those counties at some point. This is a glimpse into Nebraska and what we've experienced this past year. I'll start with a photo. The photo is just from southern Lancaster County area where we came across a field where obviously the grower had some pretty significant challenges with soybean gall midge. There's a few things in that photo You got different heights of dead plants that have occurred, some live, some still wilting and dying if you look really close. And so that just shows you the variation in response. On the other side, this 2A and 2B graphs, the top 2A figure shows you the number of larvae per plant. I think some of you asked, what's Gall Midge been doing over the years? Numbers have declined a little bit. Used to just kind of be a swath across eastern Nebraska with fairly high numbers. We're still in several counties with higher numbers, and we do see shifts between counties in terms of where we see pressure. And this past year and previous years, the previous 2 years, we started taking frequency of infested plants. And you can just see that in some places we may not have high larval number, but we do have a high frequency of plants that are infested. And so I'll spend the next couple slides. And this project, of course, funded by the Nebraska Soybean Board. I'm going to spend the next couple slides hitting some of that more basic biology, ecology type information that's useful for framing a lot of the talks that are coming. And if you're new to soybean gall midge, these are pretty important things to know. So the life cycle, this is just a basic cycle. We have eggs that are laid into the plants, and we'll talk about the fissures that are associated with that. Those eggs hatch, and we get 3 larval instars. Doctors Cook and Hodgson and I, and as well as all the students, kind of divide these into 2 big categories, really just the white or non-pigmented larvae and pigmented larvae that you see there. The pigmented is what we really use for ID. Those all occur on the plant. That 3rd instar will fall off into the soil, and then they'll emerge again as adults. And depending on when they emerge, there's different kind of interest in their generations. So where are they coming from? They're coming from last year's soybean field. I got an injured field here just north of Lincoln, Nebraska. Lots of dead and dying plants. They fall off these plants into the soil, and they overwinter in the top 1.5 inches of the soil surface. And Mika de Silva Lima did a lot of work on that during his master's degree to show they're really in that top 1.5 inches of the soil profile. They emerge in the spring. We caught them May 21st here in Nebraska, and we monitor in Nebraska, Iowa, South Dakota, and Minnesota. Then they'll move to next year's soybean. They do that shortly after emerging. And this is the first time they interface with soybean fields that are growing in the current year. There's a big difference between soybeans when they're small and they just have a single trifoliate. They don't have these critical cracks or fissures at the base of the plant that they need to infest. Those form shortly right around V2 stage. And we've got video of the female you see here with the long ovipositor. They lay those eggs right into those fissures or cracks. And that's the first time that they're really going to start infesting that plant. This next photo shows you those fissures or cracks where those eggs would be laid, and then you see that dark discoloration around that plant, and that's really what we're looking for in the field when we're trying to find this insect on various plants. And then those instars, after they hatch, they feed. That's the damaging stages that we see to soybean. You can find all 3 instars on the plant at the same time, and that's really the adult activity. After we get them into the soybeans, we get emergence of new adults, and they keep reinfesting the soybeans. We don't know really how many generations, probably 2 to 3 that can occur during the summer. It's hard to tell because of the overlap in those generations. The last part of my talk here really briefly is just about scouting. If you haven't had soybean gall midge, you don't have issues, you can wait quite late in the season till you have full pod development or even seed. And you really want to look right next to last year's soybean field in that first row or 2. That's the place where you're most likely to find it in that dark discoloration. A few key things in the landscape: if you have any dense vegetation that stands out or things that might increase the available moisture, that's the places you want to look first. And you'll hear more about that from Elliot Nell, the research project coordinator on my lab, on a mowing study that we did with Minnesota. There's those dark discolorations. These bottom 2 plants are infested. The top one just has that purple coloration we can see in some soybean varieties. And then if you peel that tissue back, that's where you'll potentially find those orange larvae underneath. And that's the diagnostic feature you're really looking for. So that's just a quick overview of some of the key questions a lot of you asked. We'll kind of dig into more detail on a lot of studies here now coming up. So any quick questions, Aaron, or we can move on?
Yeah, we had a question coming, I think from Kansas. Someone asked, any idea how many eggs a female can lay? Or do you have any idea, like, max number of larvae you've seen in a stem?
Okay, good question. I know Bob Cook's working on some stuff up in Minnesota. probably getting closer to that egg count than I could estimate based on what we've seen. The population count can vary really significantly in soybean plants. So those big soybean stems you saw, that they're kind of the end of the year survivors. Not uncommon for us to hit 200 or more larvae on those under really heavy infestations, if those are the only surviving plants out there. Typically in soybeans, if we start to exceed, and this is opinion, observation based, if we start to exceed 20 to 25 larvae per stem, we're probably pretty close to seeing dead or dying plants show up. And that's stage dependent on when they're infesting. That would be really small plants, V2 getting infested, and those numbers showing up slightly afterwards. I'll pass it to Bob if you have any comments on the eggs from observations in Minnesota.
Yeah, maybe we could slide transition, Justin.
Oh, sure.
Bob, any idea how many eggs a female could lay?
Yeah, we don't have a definitive answer yet, but we've been doing some preliminary work dissecting open gall midges and trying to figure out what are eggs in there versus just other bits and pieces of guts. And I think we're seeing probably 60, 80-ish. Don't quote me on that yet, but an individual female can have quite a few eggs, and I think with other gall midges, multiple females lay eggs on individual plants.
Yeah, it kind of complicates things, doesn't it? Yeah. All right. We had a couple more questions, but we'll save them as we get maybe closer to our first break. Thanks, Justin. Thanks, Bob. All right. Next up is our second presentation. It's Talis Rodriguez, and he's a graduate student at the University of Lincoln in Nebraska in Lincoln, sorry. And really excited to hear him talk about the impact interactions that we're seeing with deck stem borers. So take it away, Talis.
All right. So good morning, everyone. Thanks, Erin, for introducing me. I'm Talis. I'm advised by Doctors McMacken and Mangel. And today we're going to talk about soybean gall midge and deck stem borer impacts on soybean yields here in Nebraska. So first, we're going to talk about the seasonal presence of this pest. As Justin already showed you, we can find soybean gall midges in soybean plants from V2 to R7. However, Dactys has a different presence in these plants. As you can see here, they can be found from late vegetative stages until the full maturity of the plant. So for those who don't know what Dactys stem borers are, they are these grayish longhorn beetles. In this picture here, you can see a female chewing a petiole. Then it's going to lay an egg inside this petiole, and the egg's gonna hatch. The larva is gonna feed on the petiole pith, then it's gonna move to the stem pith. However, this damage doesn't cause a lot of yield loss. The big deal is later on the season when the larvae are preparing for overwinter, and they girdle the plants, hence increasing lodging and increasing yield losses. So as you can see here, we're likely to find Dactys and gall midges in soybean plants from V5 to R7. So this study was conducted last year in 10 sites across eastern and south central Nebraska. And Kennesaw was the single site that was irrigated. This study was placed on the field edges where the presence and impacts of Dactys and gall midge is greatest, and had 5 treatments. So we had our untreated, which we just planted the soybean seeds and had no interventions in those plots. We had disease management with seed treatments and foliar applications of fungicides, but we won't discuss that for the sake of this presentation. That's not our objective here today. We had fipronil seed treatment, which is a very effective treatment against deck stem borer. We had healing, which is covering those fissures on the lower stem of the soybean plant. And it's a very effective tactic to control gall midges. And we combine the both last treatments together. So here you can see some of more like a visual result of our treatments. On our first wrap that's located in the edge, we can clearly see that hill plots, they have healthier, it's greener. We have better plants there. And we can see the impact of gall midge on the fissure exposed plots. On the second wrap, we can still see the difference between hilled and non-hilled plots. And on the third plot, that's not so contrasting. So let's start talking about soybean gall midge impact. So for this comparison, we were comparing hilled against not hilled plots. As you can see here in this picture again, a very effective method against gall midge. So across these 10 sites, we ended up excluding some of them. We're not going to talk about Kennesaw results. We had no gall midge in that site. And we also lost Skull and Hall sites because of herbicide application. So we're going to toss them out. So on our 7 remaining sites, we had 2 sites that we had some problems. One of them was flooded and the other one has no source for gall midge. And our results on these plots ended up that not hilled plots yielding more than hilled plots. However, on the other plots, we see huge impacts, ranging from 12 to almost 35 bushels per acre loss in this plot, in the sites. How many larvae per stem we found in those sites? So on the low impact, low gall midge impact sites, we found no more than 2 larvae per stem, for soybean stem. And oops, yeah, it was a technical issue. Let's keep going. So on the low impact sites, we had no more than 2 larvae per plant. However, on the more impacted sites, we had way more larvae on these plants, ranging from 5 to almost 20 larvae per plant. Now we're going to talk about Dextin borer. So for this comparison, we are contrasting fipronil treated and our check plots. And it's important to highlight that fipronil has no effect on gall midge. So we found a lot of gall midge larvae on both of these plots. So in 4 of the 7 remaining sites, we had our check plots yielding more than our fipronil treated plots, around 0, like no difference at all, to +7 bushels per acre. And on the sites that we had some impact of Dactys, we didn't have like huge losses like we had for gall midge. So on the sites that we had good control of Dactys, the control was ranging between 65 to 90% of larvae control. And on the yield losses side, on sites that we got yield losses, the control was ranging between 65 to 75%. Now, we're going to compare hilled and fipronil treated plots against untreated plots. So as you can see here, we had a good reduction on gall midge presence in these plants. And we had the same sites that were flooded or we had no SARS. The untreated ended up yielding more than our fipronil hill plots. And we got similar yield losses on the other sites. When we compare, so we can see here that when you have Dactys and gall midge, Dactys doesn't increase, doesn't have a significant impact when we have Dactys and gall midge together. And then we decided to compare these fipronil and hill plots against hill plots. So productivity again between these plots were very similar, ranging between 55 bushels per acre. And as you can see here on plots, we had most of the impact was caused by gall midge. We don't see very different yield differences between these plots. So about past interactions in the literature, we couldn't find any Dectis or soybean gall midge paper associated with diseases. However, we found gall midge and Dectis associations in plots that gall midge presence was really high. And in the literature, we have a similar species to gall midge, that's raspberry cane midge, that's associated with a fungus causing this disease here that's known as midge blight. So the summary for this presentation is that gall midge had a great impact on 5 of the 7 sites that we're analyzing. Dectis only had impact in 3 sites. When you have both paths, we see that soybean gall midge impact is prevalent. And we also found some interactions in plots that gall midge presence was pretty high. So I'd like to acknowledge my community members for guiding me, my lab members for helping me with collecting all this data, and extension educators for connecting us with the growers. So if you have any questions, I'll do my best to answer them. Thank you.
Thanks, Dallas. We're a little short on time because we just have kind of like these short bursts of updates. So I think we'll save questions about Dectis and midge interactions for our first break at 10:45. So
Okay.
if you could, yeah, stop sharing, and Sarah will get going with the next one here. Next up is Sarah Von Gries. She's a graduate student at the University of Minnesota, and her project is an excellent example of a multi-state effort, and she's going to be talking about parasitoids and predators, our natural enemies that we're finding for soybean gall midge. And we see your slides. We see you. Go for it.
All right, thank you, Erin. All right, so I'm going to be talking about parasitoids of soybean gall midge. So first off, what is a parasitoid? It's an insect that lays its egg inside of a host, and then it will feed on the entire host until it emerges as an adult. Our lab in Minnesota has previously identified a wasp known to parasitize SGM in Minnesota, and this new species is known as Sinopius maximum. A previous student in our lab found low parasitism rates in Minnesota fields with the highest daily rate of 10%. So this is the percent of SGM larvae collected on one day that were parasitized. And then the seasonal rate was less than 2%, so not very high. Since then, we have expanded our exploration of parasitoids to include South Dakota, Iowa, and Nebraska on top of Minnesota. And to do this, we collected infested stems and placed them in emergence buckets to rear out adult insects looking for parasitoids as well as adult SGM. And in the buckets from Nebraska, we found 23 parasitoids looking like this, which is different than Sinopius maximum we found in Minnesota. Here's a photo of this wasp found in the emergence cages in Nebraska. All 23 of them were female. So now we have this new wasp that seems to be associated with SGM, but before determining if it is a parasitoid of SGM, we first need to identify this wasp. With the help of taxonomists, we were able to identify this parasitoid. So next I'm going to walk you through our process using molecular tools and physical traits to find other records of this parasitoid. We used molecular tools to get the DNA sequence of the adult wasps that we collected. We then compared the sequences to DNA databases where 2 DNA matches were found. One of them was collected in 2014 in Quebec, Canada, and the other in 2017 in Ontario, Canada. Luckily, these specimens were deposited as vouchers, and one even included a photo from 2014. So taxonomists ordered the voucher and compared it to our specimens using physical traits. So here are the 3 photos showing the 3 specimens from 2014 through 2021. All physical traits were identical. After some digging, one of the taxonomists came across a wasp that looked very similar to ours called Sinopius ruficoxum. This description was based on one specimen from New Brunswick, which is close to those other Canadian specimens. The taxonomist tracked down the single specimen and then compared it to ours using those physical traits. And it was identified to be the same species we were working with, named Sinopius ruficoxum. So we have identified this wasp. Here's a map showing the current known distribution of Sinopius ruficoxum adults compared to their distribution to the known counties with SGM infestation. You can see the first 3 records found this wasp nowhere near SGM, so maybe it has some other host, or maybe SGM can actually be found in those areas and never has been before. Here are side-by-side pictures of the 2 parasitoids. Notice how Sinopius ruficoxum has a long, skinny abdomen in comparison to maximum. So we know maximum has a low parasitism rate, but what about Sinopius ruficoxum? So this leads me to my 2 objectives for today. The first, to determine if Sinopius ruficoxum is indeed a parasitoid of SGM, or if it just happened to emerge in that emergence cage. And 2, to characterize SGM parasitoids spatially and temporally. So sampling occurred in 2023 in 4 states. There were 17 fields that we sampled every 2 weeks. So the number of samplings varied for each field, depending on the field. We collected 40 random stems on the field edge within a 50-meter distance, and then dissected out the larvae and preserved them in alcohol for use in molecular methods. Then we screened the SGM larvae for wasp DNA. So we used molecular tools to detect the presence or the absence of wasp DNA, either of the species, in SGM larvae. And we did this to see which larvae were parasitized by either of the wasps. Moving on to the results. We were able to detect Sinopius ruficoxum DNA in SGM larvae, which tells us that Sinopius ruficoxum is indeed a parasitoid of SGM. Additionally, there were 18 individual larvae that screened positive for both Sinopius species, showing that multiparasitism is possible. Next, I'm going to show you the highest daily percent parasitism detected for each state. And the highest daily percent parasitism, again, is that highest percent of the larvae parasitized on a given day. So for Minnesota, the highest daily percent parasitism was 10% for Sinopius maximum and 18% for ruficoxum. For Iowa, it was 7.5% for maximum and 2.5% for ruficoxum. For South Dakota, it was 14% for Sinopius maximum and 1.5% for Sinopius ruficoxum. And for Nebraska, it was 22% for Sinopius maximum and 60% for Sinopius ruficoxum. So we see the highest level for both parasitoids was in Nebraska, and just how much higher parasitism was for Sinopius ruficoxum in Nebraska compared to all the other states. Despite the state differences, both Sinopius species were detected in all 4 states assessed. Here's a figure that will combine all of the fields in all of the states where parasitism was detected to look at the detection of both parasitoids temporally. So on the x-axis, we have our month of collection, and on the y-axis, we have the cumulative percent larvae parasitized, which is the percent of the population attacked by the wasp that is destined to die. And then the color will show the parasitism type, either maximum or ruficoxum. So here are the results. And if we take a look at Sinopius maximum, which is in gray, we see parasitism is detected in June, and then August and September. So in the early part of the season and the late part of the season. Then for Sinopius ruficoxum, which is in orange, we see parasitism is detected throughout the season, but there's peak detection in the middle of the season in the month of July. So if we put these together, we see that Sinopius ruficoxum seems to fill the gap where Sinopius maximum parasitism is not present. So maybe Sinopius ruficoxum has filled the niche where Sinopius maximum couldn't. So this shows potential for both parasitoids to be contributing together to biological control of SGM. In summary, Sinopius ruficoxum has been found in other areas outside of SGM's known range. Sinopius ruficoxum is a confirmed parasitoid of SGM. We did find multiparasitism in the field. We detected the highest percent larvae parasitized in Nebraska with up to 60% for Sinopius ruficoxum. and the parasitoids seem to complement each other temporally. These results help us to better understand the parasitoids of SGM, which then informs the development of IPM programs, with Sinopius ruficoxum maybe having a higher potential as a biological control agent, but they could also work together throughout the season. And with that, I thank the following, and if there's time for questions, I will take them now.
Great. Thanks, Sarah. As we transition slides, we had a question about the host range for these two species. Do you know if they would be impacting other types of insects or pest insects?
Yeah, great question. So this genus Sinopius, so the specific group of parasitoids, are known to be very specific for their hosts. So they're very specialized. So generally that's just 1 to 2 hosts.
Okay.
So it's most likely that they don't have other hosts, that they're only impacting soybean gall midge. But with ruficoxum being found in Canada and other areas outside of SGM's range, that kind of tells us there's probably something else out there. If not, maybe soybean gall midge was actually found in Canada as well, and we just haven't detected it.
Yeah. Oh, interesting. Thanks, Sarah. Appreciate the updates. A lot of blood, sweat, and a few tears, literal, for this project. So yeah. All right. We are moving on. We have one more presentation before we take a panel break and try and catch up with some questions that you guys have been sending in. Keep it coming. We have Bob Cook. He works at the University of Minnesota, and he's stepping in for his student, Phelan, who couldn't be here today.
Yeah, so I'll do my best to fill in for Phelan. He regrets not being able to join us today, but he got ill last night, and I'm going to be presenting some preliminary results here on a new line of research related to what we call entomopathogenic fungi, which is basically just kinds of fungi that attack and kill insects. See if my slides will advance. Okay, so Justin provided an overview of the life cycle of soybean gall midge, so we're not going to dwell on that. But what I want to highlight is that a large portion of the gall midge's life cycle, the individuals are kind of hidden or protected, either within the plants or in the soil, which can make it challenging to manage this pest. However, one option that, like I said, that we're exploring is with these entomopathogenic fungi. They're known to attack and kill multiple different kinds of insects. You can see a beetle here that's been infected, and you can see all the white fuzzy mycelia that's resulted from the infection by this fungus. And the way these fungi work is they're going to release spores, and then those spores will land on the cuticle or the outside the insect. That spore will germinate, and then the fungus will start growing in and throughout the insect, eventually killing it. And then the life cycle will continue as that fungus will release new spores to attack other insects. So the thought for soybean gall midge is that hopefully these fungi will maybe be able to provide a better tool or an additional tool for accessing this pest in those hidden locations, either inside the plant or inside the soil. Another promising aspect in regards to this is that in Europe, certain kinds of entomopathogenic fungi have been shown to attack the raspberry cane midge, which is a closely related species of gall midge, closely related to soybean gall midge. So just kind of a quick recap of why we're looking at these fungi. In general, they have fewer non-target impacts compared to use of insecticides. In some cases, they can establish and grow inside of the plants and inside the soil. And these haven't yet been evaluated in specific for soybean gall midge. So for Phelan's work, the aim was to perform an initial screening using several different strains of fungi to see if they have potential for biological control. So what we did is went to Southwest Minnesota, collected soybean plants that were infested with soybean gall midge, brought them back to the laboratory, and we started a laboratory colony of the insect. And then from those plants that we had in the laboratory, we dissected those open to attain those large mature larvae that Justin was explaining in his presentation that are kind of indicated by that dark orange coloration. And then for the fungi, we teamed up with Dr. Bob Blanchette from the University of Minnesota in the plant pathology department. And he's been doing a lot of work with biological control of the emerald ash borer. And they've obtained different strains of fungi from trees that were infected by the emerald ash borer. And our interest here is that these fungi from trees in Minnesota are adapted to Minnesota's climatic conditions. And then working with Dr. Blanchette's team, we created an inoculum, which is basically spores of these different kinds of fungi in a liquid solution that was then sprayed on soybean gall midge larvae placed inside of petri dishes. So this is kind of a worst case assessment from the gall midge perspective, right? You're in a petri dish and you're essentially getting doused with a solution containing these fungal spores. We then allowed those dishes to dry and then transferred the larvae into vials of sand. And we had 10 larvae per vial, 5 vials per treatment. And then stored those vials in a growth chamber under conditions that we know are conducive for survival of the soybean gall midge. And then after 12 days, we checked those vials to determine if the gall midge were infected by the fungi. And the terminology that the mycologists use is mycosis or mycost, which means that the insect is dead and with visible signs of fungal infection. So you can see a picture of that here, a gall midge larva that's been killed by a fungus, and you can see the fungal mycelia growing out of it. The treatments that Phelan used were an untreated control, and then a couple strains of Bavaria, one that came from the emerald ash borer or EAB galleries. The other is a commercial strain of Bavaria, and then Purpureocilium, Clonostachys, and Metarhizium are different species of fungi that Dr. Blanchette's lab obtained from those emerald ash borer galleries. This graph shows some of Phelan's data, where across the bottom of the graph, we've got the proportion of insects that were dead and infected or mycosed due to these different kinds of fungi. And then those strains of fungi are listed on the side of the graph. For the control, Phelan found that none of the insects were mycosed. For the Bavaria strain that came from the emerald ash borer galleries, we found nearly 75% of the individuals were mycosed, which again means they were dead and infected with the fungus. And then intermediate levels for all those other strains. So this is promising because it shows that all of these fungal strains can infect and kill soybean gall midge. And then based on the stats, we saw that the Bavaria from the emerald ash borer galleries had higher levels of mortality or mycosis compared to the control and several other fungal strains. So just a quick recap here of the results and some of the implications. Again, each of these fungal strains was able to infect and kill soybean gall midge. That Bavaria strain from the emerald ash borer galleries was the most effective, again, killing about 74% of the larvae, and just emphasizing again that was a Minnesota collected strain, so we know it's adapted to Minnesota conditions. However, there's a lot more research to be done yet before this can really be advanced as an additional management tactic, right? We did kind of a worst case scenario in Petri dishes. Our next steps are to evaluate this under more realistic conditions, actually with plants and soil. And then we'd like to evaluate some of the commercial products or formulations of some of these different fungal species like Bavaria and Metarhizium. So with that, just like to thank the support from the Minnesota soybean growers for funding some of Phelan's work. And then, a shout out to the team in my lab that's helped dissect and count tons and tons of gall midge larvae. So hopefully I didn't throw us off too much on time here.
Oh, you did great. Thanks, Bob. I know it's not easy to cover someone else's project with like 30 minutes notice. So yeah, thank you to everyone who's sending in questions. I've been trying to collect them, and we have a good amount of time now to try and answer some of the questions about the updates that were presented with the first 4 folks. And as you could imagine, Justin, there's lots of questions about movement of the adults, I think within fields and between fields. Do we know how far they can move? And someone said, when are they getting to Wisconsin?
I left my crystal ball at home to know when they get to other states. I don't know if any of you brought yours. So that's a tough question to answer. But we have a lot of great colleagues in those states that are looking and keeping an eye on this. And I was actually just in Wisconsin giving a talk. So I'm assuming if it's there, they'll pick it up, but there's certainly no signs of a lot of efforts of surveying. And so people can kind of rest easy knowing that there's people looking. As far as movement, I just typed briefly in there, we detect them at 500 feet from an overwintering source. So they, can go that far because there would be no other location nearby that we are aware of that they could come from. And then in surveys in Nebraska, where we've come to areas where we found out there was no soybeans around that for 1.5 miles or 2 miles, we've detected them in the center of those zones. That caveat being that sweet clover and other sources, we can't account for all those. So I would say confidently 500 feet. That was our maximum distance taken. But they're not great flyers. We all kind of know that from what we've seen.
Yeah, and just with the distribution map that you showed at the very beginning, it's now a few counties north, east, west that we had last year. I would anticipate that to continue, but knowing that we're looking very, very hard on those like uninfested counties, we're looking at lots and lots of plants, as you said. So it's presence, absence. It doesn't necessarily mean economic populations and kind of those fringe counties. Yeah. I'm wondering if you've had any thoughts about the overwintering that we've been experiencing in these states as far as, success based on temperature, residue, snowfall. Everyone wants to know if they're going to make it this winter.
Well, Bob's got some nice data. Yeah, go ahead, Bob, on the supercooling.
Yeah.
So that's another part of Phelan's work. And I think he presented on that in one of the previous webinars. So there should be a recording of that. And some of our earlier work was looking at short-term exposure, so exposing the larvae or the cocoon larvae, which is the overwintering stage, to different low temperatures. And from that, they seemed pretty resilient. Like they could do a pretty darn good job surviving winter conditions. But some of Phelan's follow-up work now is looking at exposing those gall midges to longer durations of these cold temperatures, which is going to be, a more realistic scenario. And we're seeing that they're certainly not surviving as well when they're exposed to things for longer periods of time. So I can't give a definitive prediction yet on, okay, we experience these kinds of temperatures, we're going to see this much mortality, but we're hoping to move things in that direction. Phelan wants to repeat some of that research this coming fall and winter, looking at longer term exposures. And then, a next step from a couple of years of data like that will be to, maybe do some modeling based on soil temperatures to be able to try to make some of those predictions like we can do with some other insects. Like Aaron's lab, annually puts out, updates on bean leaf beetle mortality over the winter.
Yeah, I think some of our northern counties in Iowa have had soil temps that are like 19 to 22. So it does seem like it's a colder winter as far as like in the soil compared to last year. And so everyone wanted to know if like all the rootworms are dying, and then, now soybean gall midge. And so maybe just keep that in your mind as far as, I assume Minnesota's colder than Iowa, but they seem fairly cold hardy. Justin, can you say with any confidence if gall midge can be passed through like seed production? Yeah.
I don't know. I early on when this broke out in 2018 and 19, we got questions from APHIS in terms of potential seed movement. And I know each one of us has probably observed this and came out and attached instead, but I know I've seen photos from either one of you that show them in the pods. In some rare cases, you'll find gall midge up there, especially an injured pod. But to survive on dry seed after harvest and all that, highly, highly unlikely.
What about on equipment, like planting from field to field? Any chance of movement between fields on like machinery?
Again, if we're looking at when they'd show up and if we're spraying fields where we might be in, they're really falling to soil. So you'd be running over them. There's very low chances of that possibility. You might be able to pick up that soil and move them with the soil, but there's probably a greater likelihood they're already out in those other areas because of adult movement than moving vast quantities of distance with soil. I don't know, Bob or Aaron, if you have thoughts on top of that.
No, there's a lot of interest because I think they demonstrated that happens with soybean cyst nematode. And so that the sanitation is really important, but I totally agree with you about just the biology of the pest itself and
Yeah. I mean, I would clean off my sprayer after spraying a field if I'm in soybean gall, which just get the chunks of mud off of there. If they're in something, that would be it. I know commercial, like working with industry when they were here, they washed their pickups before they left. They were also headed back to Illinois and certainly didn't want to be any starters of anything.
Yeah.
So extra precautions, not a bad thing if you're heading into an area where it hasn't been.
I think for the life stages in the soil, I think something like soybean cyst nematode is probably much more resilient than the life stages of soybean gall midge that are in the soil. I think our general feeling is that gall midge is pretty sensitive to moisture levels, humidity, things like that.
Okay. Yeah, great. Bob, I think it may be fair to say that Minnesota has more dry beans than maybe other states. Can you comment on if they're impacting dry bean production at all?
Yeah, so Bruce Potter did an interesting study several years ago, and I think some of that was shared in an earlier webinar where he put potted plants of different bean and legume species out into fields, and he did get some oviposition and larval development on some dry beans. And then with Bruce and some of my students following up on it, detected gall midge infestation in actual dry bean fields in Minnesota, and then confirmed that it was soybean gall midge. The injury to the plants was fairly similar to what we're seeing in soybean, some of it more up in the axils, but it seemed like lower levels of infestation. But this is something where we've been able to secure some funding here in Minnesota to follow up on, to actually look at how susceptible or attractive these different dry bean and other legumes are compared to soybean.
I think Bruce he's here on the webinar and he's asking his very Potter-risque questions. So he's still continuing lifelong learning. So shout out to Bruce. Thanks for joining us today. We have a few questions that I'm hoping Sarah can answer when it comes to the wasp that you talked about today. Do you think that the rate, do we know anything about the density dependence of the wasp and the midge? Is there a relationship there?
So we don't know about the density in the overall area, but we were able to, I believe it was last year, I'm just pulling that up right now, within stems, so stems with higher density of SGM are more likely to have parasitism present, but have a lower proportion of SGM parasitized. My guess is probably due to competition. The parasitoids probably realize there's a lot in the stem and they don't want to put all their eggs in one basket kind of thing. So that's what we know so far. But based upon what we've seen with parasitism percentages between the states, Nebraska definitely has the highest and they have most infestation. So maybe overall there could be something there, but we haven't tested that specifically.
It's not a competition, but they seem to be winning. All
But
right.
some of this will be coming out of Sarah's work now that she's got all this data from these multiple states, looking at some of these spatial temporal dynamics and density dependence and things like that. So stay
Yeah. Is there anything in the literature for these wasps, or maybe wasps in the genera, that we know about their life cycle? Like if we wanted to encourage the wasps to maybe establish in an area with midges, what can we do to help them?
Yeah, so I haven't seen anything about this specific genus, but it has been shown that nectar, sometimes some kind of pollen or nectar sugar sources, can be a good resource to let the wasps live longer.
Mhm.
I haven't seen anything specific about it, but my guess is that it's possible. We would just have to test that out
Yeah.
to see if those resources would help.
I think
Similar
another
to
big
other.
thing is,
Go ahead, Bob.
I think another big thing is pesticide use
Mhm.
in general. These various kinds of parasitic wasps like this are more susceptible. They're easier to kill with insecticides than the pests. So, anytime we're spraying the fields for gall midge or aphids or whatever, we're likely taking out, removing these beneficial wasps, so just a little more incentive, I think, to be using thresholds, make sure we're using the insecticides when we really need to, right?
And I would say stay tuned as well, because with all these states that we've been assessing, we are going to try to do some kind of landscape analysis to see if any forested areas or grass areas could impact where we're finding high parasitism rates.
Okay, yeah, I was assuming that it would be water nectar feeding as adults.
Mhm.
So if you have those resources around fields, maybe that would be a little bit of a carrot for them. And I also, like Bob, I would assume they'd be highly susceptible to insecticides. And so it's hard to have a situation where you're relying on insecticides for whatever reason and then expect parasitism to be also a suppression strategy. So you can't, it's hard to have both. Yeah.
Yeah,
Great.
the good thing though, Aaron, is some of these newer chemistries that are coming out, like Safina, sulfoxaflor, things like that, they are
Mhm.
narrower spectrum, right? Where they still do a good job killing certain pests, but they're less toxic.
Yes.
Some of these good insects.
Mm-hmm.
So
Yeah, especially
there are improvements beyond the older broad spectrum insecticides.
yeah, definitely narrow target. Like if soybean aphid is your primary pest, maybe you're not necessarily just thinking about soybean gall midge, but yeah, that's true. Some of the newer products would be a good fit. All right, thanks, Sarah. I'm going back to, many of you are asking questions about tilling and hilling, and I think we're going to save those for the second panel discussion, because I think some of those questions will be answered by a few of the presenters. Let's see. We had a question about the pressure seemed a bit lower, and you had mentioned that, Justin, sort of in the introduction. Is it related to rainfall? Like what can we say about maybe why the numbers are ups and downs?
Yeah, I saw that question. I jotted down a few things. It's a complicated one. We've done some rainfall studies in Nebraska. Most of those studies kind of point to increased adult emergence if we have additional rainfall versus nearby areas with similar populations, we see less. So rainfall can increase adult emergence. It's not wild in its difference between those two. Sometimes it's just numerical differences, but we have had some significant differences. When we have heavy rainfall, especially in the spring, sometimes what happens is we get delayed plantings, and Natasha Umeziu, who's on here, be talking about some bioassays later. That delayed planting can reduce infestations as well. But for an individual grower going like, hey, 2023 things were bad, 2024 they were lower, hang on for the host plant resistance discussion. You may be changing varieties, and that may be part of what's happening. Think about the source fields you had nearby. Did you have a corn or soybeans go to corn rotation, where maybe you didn't have that adjacent source anymore and the pressure dropped because of that. My last little note to add on this is that wet dry, if we start wet and we get gall midge in there and it seems doing well, things get dry later in the season, it adds an additional pressure on that plant because of the feeding on that xylem and phloem area where the water and nutrients are moving through. Additional environmental stress can accelerate plant death. And Mitchell Helton, Aaron, your student that, you know, I was on the committee for, we got data from here. It wasn't hard in Nebraska. We got hot, dry weather. Those injury scores really climb quickly. So sometimes late season dry weather, opposite effect in terms of impacting stuff. It can really impact it quickly.
Yeah, the erratic kind of hot, dry, hot, or wet, dry, all that. I think the stress can make the feeding that is there worse. Yeah, with some of the like lodging, but like creating really brittle stems. Yeah, thanks, Justin. That's great. Dallas, I was wondering if you didn't know if you had DECDs or not, which I think a lot of people in Iowa may not realize that they have DECDs kind of on the western side of the state. When would be a good time to just check presence absence?
Okay, can you hear me?
Yeah.
So when we should scout them? Well, we were monitoring them using the corn rootworm cages. Last year, we found the first adult by June 10th. So
Okay.
that's one way to monitor them. But on plants, it's easier to find them later in the season when the larvae already reached the third instar and they moved from the petiole to the main stem or thicker branch. However, I mean, you can find out that they are there, but it's too late to do anything to control them.
Yeah.
And yeah, I think, but you were asking about the northern part of the state.
I think there are DECDs along some of the western counties of Iowa, but people aren't used to looking for them and may not realize that they might have some lodge plants and, you know, I think they might associate it with soybean gall midge. But I think just as your research showed, you could have both at the same time. And so on those kind of areas where the populations could overlap, it sounds like maybe August, like if you didn't know if you had either, you could scout in August and maybe you could be looking for both and some of the
Yes.
visual symptoms and like the lodging and things, the wilt and all that would be apparent if you had DECDs and or midges. Would that be a safe assumption, Justin and Dallas?
Yes.
Okay.
Yeah. I think Tully's answered it really well. Yeah, by August, we start to see those wilting or dead petioles. Once you start seeing those along fields, following those into the plant is the best way to know if you have DECD stem borer.
Okay. I think for at least our weather experts are saying it's going to be a cool, wet spring and maybe kind of staggered planting dates. Do you have any idea if like really saturated soils could impact larvae and pupae that are kind of trying to come back to life after a cold winter and turn into adults? Is there like a suffocation point like you might see for other soil dwelling insects?
We really, a lot of us dislike soybean gall midge. Like, can they suffocate? How many ways can they die? So we had flooding in 2019 at the Eastern Nebraska Research Extension Education Center. Areas where we placed cages were underwater for 7 days, consecutive days. And we got soybean gall midge adult emergence from those cages. So it's one observation, but you know, they didn't seem to be dampened in terms of emergence by that amount of water on them for that amount of period.
Hey,
Okay.
Justin,
Yeah.
to follow up on that, we've got some laboratory indications that they can survive a long time in water.
Okay.
We've taken larvae out of stems and put them into vials with water, and then after certain amounts of time, you know, put them back into conditions that should allow them to continue development, and yeah, much longer than a week, they're surviving just fine.
Oh, okay.
Thanks, Bob.
You know, in the field, things might be different, right? You know, where there could be other, you know, like microbial activity that's maybe decreasing oxygen content in the water, whereas these were just vials and we weren't measuring how much oxygen is in there. You know, there's been some detailed work done on corn rootworm with that kind of stuff, you know, where it'd be pretty cool to follow up with soybean gall midge, you know, with more controlled studies to look at this.
Yeah. So maybe cold, wet, wouldn't expect it to impact their survival too much, you know? Okay, good. Well, thank you, everyone. All the speakers in the first session, we got through a lot of questions. I think we'll move on to the second set of presenters. We have four more updates before we wrap up with another just kind of general panel discussion. Next up is Natasha Umezu. She's a graduate student from the University of Nebraska in Lincoln. She's this close to getting her PhD, and we're very excited for her. She's done a lot of great work with soybean gall midge over the last couple years. And today she's going to be talking about some of the bioassays that she's been doing in the McMecken lab. So you're ready. Yep, go ahead.
Okay, thanks, Ari. So good morning, everyone. Today I will be presenting the bioassay that we developed for soybean gall midge. And okay, so to start my presentation, first I want to explain a little bit what would be an insecticide bioassay for those that maybe doesn't know. A bioassay is a controlled experiment that is used to evaluate the effectiveness of active ingredient and access resistance development. So with this, we can determine if an insecticide could be toxicity and efficacy for a specific pest. And in this way, we can identify a lethal dose that could help to correlate the amount of insecticide that can be used in the field. So maybe some farmers today in this webinar are thinking, so what would be the importance of a bioassay for them, right? Because a bioassay is not a practice that they can do in the field. It's not something that is practical for them. But to answer this question, I want to bring back some videos from our Soybean Gall Midge webinar that we presented a couple years ago. And for those that didn't watch, you can go to our website and you can find there our other videos, so you can go there after you watch this webinar. But in the previous webinar, we presented my planting date and seed treatment that Justin already discussed it here a little bit before today. But just to give you a brief overall about this research related to the seed treatment, we did a field research conducted in 2021 and 2023 in Eastern Nebraska. And besides we evaluated the planting date, we also tested seed treatment. And we used a neonicotinoid that was imidacloprid. And in 2021, we used a rate of 0.12. In 2023, we used the rate 0.23. So we increased the rate for 2023. And when we look the results from both of the year, you can see that actually statistically, we didn't have a difference between the treatments. But if we look the results in the numerical results, actually we can see that in 2021, we had, I will take the IUSPC result, it means the injury that was caused from soybean gall midge to the plant. So higher the IUSPC, more the plant were injured. So in 2021, the untreated showed a higher injury. But then, if we look the results from 2023, it shifted. So actually the seed treatment showed more injury compared to the untreated. So since we had this numerical difference within the years, and well, we are scientists, right? So scientists, we are curious, we want to know, we want to explore more the findings that we had. So we want to understand why we had this difference within the years. And when we talk about seed treatment, it is important to consider that there are other factors that can impact in the efficiency of the seed treatment. So we don't know how much of the insecticide can reach the insect or how long the insecticide can last in the plant, and also the environmental conditions, if this can impact in the efficiency of the seed treatment. So we thought, well, if we remove all those external factors that can impact on the seed treatment, could we test and see if the imidacloprid is actually effective against soybean gall midge? So to answer this, we developed a bioassay, and then we could determine if the soybean gall midge is susceptible or not to imidacloprid. So in 2024, we went to the same location where we did our planting date and seed treatment research. And in those locations, we collected infested soybean stems with the soybean gall midge larvae, and then we placed those stems in a paper bag, and then we put in a cooler to avoid larvae mortality, and we brought those stems to the lab. And then to set up the bioassay, we used the imidacloprid at a 99.6% of purity, and we tested 7 concentration range that came from 0, that was our control, until 160 micrograms per microliter. And the control, we treated with double distilled water. So here in this image, in this picture, I just want to tell you and show you that we did a serial dilution. That it means that from the highest concentration that we wanted to test, we diluted in distilled water until we got the lowest concentration. And to set up the contact bioassay, in the Petri dish, we placed 2 filter papers. So we placed 1 filter paper on the top of the Petri dish and in the bottom of the Petri dishes. And then we pipette the concentration in each part of the Petri dish. And the reason that we did this is because the soybean gall midge larvae, actually they jump. So we want to make sure that the larvae would be exposed all the time with the insecticide. And then after we transferred 10 orange larvae to each Petri dish, and after 24 hours and 48 hours, we assess the larvae mortality. So here in this first picture, you can see the larvae when they were alive. They had the same shape. They look healthy and nice. But then in the second picture, you can see when they are when they were toxic for the insecticide, when they are dead. So they had this like C shape. And in this video, I hope it's showing, it's freezing a little bit, but it's showing they also show this behave when they were being toxic for the insecticide. So they were like shaking. In this situation, I didn't count as dead because I was not sure if they would recover or not. But then after 48 hours, all of those were dead. Okay. So the results in the graphs that I'm going to show for the results, in the y-axis, we will have the percentage of mortality, and the x-axis will be the concentrations. And as expected, the mortality increased as the larvae were more exposed to a higher concentration. And the LC50, that is the lethal concentration that can kill 50% of the larvae, varied depending on the location and also the timing of exposure. So when we look the results per location, this is for Saunders location. The LC50 for 24 hours of assessment was higher than when we compare it to the LC50 for 48 hours. So it means that we need more insecticide to kill 50% of the larvae in a shorter period of time. And the result was the same for Lancaster location, where the LC50 for 24 hours was higher than for 48 hours. So in the end, we could answer the question that if imidacloprid would be effective against soybean gall midge or not. And yes, we could see that imidacloprid can be toxic for orange larvae, because from the concentration range that we tested, we found a concentration that could kill 50% and also 100% of the larvae. But it's important to keep in mind that the lethal concentration varied between the locations and time after the infestation. So after we answered this question that, okay, imidacloprid can be toxic for gall midge, so why in the field we are still having those differences, right? And then we want to understand how much of the insecticide can reach the insect. So then last year, we did a preliminary study where we collected soybean stem that had the seed treatment, and then we separate the stem in 2 parts. The part that was more inside, and then another part that is more like outside of the plant. And we sent those tissues to the laboratory, and we saw that the amount of imidacloprid from each part of the stem, that is a huge variation of the concentration. But again, this is still something that we are starting, and we are still analyzing the data that we can correlate with the bioassay. So stay tuned, because maybe next year for the webinar, we will have more data for this. With that, I want to thank you, my lab, and all my committee members to help me to develop all the research. And I will be happy to answer questions if we have time.
Thanks, Natasha. Yeah, great. I can't wait to see more data coming out from this project. I think we will transition to our next speaker if just so that we can keep on time, but we'll have another panel discussion at 12:45 p.m. So while George is setting up, just a quick introduction for those that don't know Dr. George Graff. He's a professor of soybean breeding. If you didn't know, he's a pretty big deal at the University of Nebraska in Lincoln. And it's another example of like a long-standing multi-state project between the groups today. And just excited to hear about some of the genetics, the updates that you've processed from the last couple seasons. So time, George. Yep, take it away.
All right. Thank you. All right. This is about looking at the genetics of host plant reaction to soybean gall midge. And we purposely didn't call it resistance, although what you'll see from this is that there is resistance. It's just a little bit complicated. So I wanted to just take a second to acknowledge the people. Luis Posadas is a research assistant professor with our group, and Aaron Hoagland's a field research technologist. And then the best thing about gall midge is get to work with all these great entomologists, Justin and his group, and Aaron and her group, and then Adam in South Dakota. So I really appreciate being able to work with the group and get make some headway on this issue here, especially from the host side. Just a quick history. This has been going on for a few years. And so the first thing we did was a large scale screening, because, we wanted to know, is there any potential for any kind of differential response in soybeans? And so we had a group of over 700 accessions from the USDA soybean germplasm collection. And that was actually came from another North Central funded project where we were working with, looking at better use of the collection, especially for complex traits like yield. And so we knew the sample that we had represented all the genetic variability that was in the 20,000 accessions of the collection in this group of 700. It was a lot of work. I think Justin finally recovered, his back finally recovered from those couple of years, and the other people taking all the notes on the field work. But we were able, in each year, evaluated 2 reps, 3 locations, larval presence and absence. We couldn't do a lot of detailed studies, but it was really, like I said, initial screening and looking at injury scores. And what we found was, this is kind of looking at, we did find individual QTLs or, genomic regions that were significant, but when we looked at different numbers, like took the 10 most significant QTLs or the 20 most significant or 30, there was really, you can see there's really no correlation between the injury, which is on the on the y-axis and the index. So and the number of genes really doesn't show any kind of correlation with genotype and phenotype, let's say, don't really show any correlation until we get to 5,000 or more markers. So the resistance is multigenic and it's quantitative. We need to look at genes throughout the genome and to be able to have any kind of decent correlation with the phenotype. So it's kind of tells us maybe there isn't a single resistance gene, but it's also kind of interesting and presents other opportunities. So what we did was use those 20,000 genes, or actually all the genes that were polymorphic, so a little more than that, and go back to the collection because the USDA soybean collection has genotype information on every accession of 50,000 SNPs information. So we were able to, with that correlation between the genotype and phenotype, project that onto the genotype information that's available in the collection, and from that, select the 100 accessions that were predicted to be the most resistant, or we actually took 90 that were the most resistant and 10 of the most susceptible to kind of test and validate what we were finding in those first couple of years of screening. And then after a year of seed increase on those new lines, we evaluated those in infested sites, got injury scores and area under the severity progress curve. And we also started evaluating elite lines, at least from our soybean breeding program, to kind of look at what was available. Is there anything in elite lines? And so what we're able to see now over years is that we see some consistencies. There's differential response, differences in number of larvae per plant, and differences in plant response to infestation at individual locations and over locations. We see from the 23, 22, and 23 tests that when we add plant injury scores, the AUSPC on the y-axis and the accessions along the x-axis, and overlay the larval counts on that. There is larval presence or different numbers of larvae present, and we still see differential response with really low, very low injury scores or zero, a lot in a few of the accessions, to very high. And so we always included a susceptible check, a commercial check that was present from the first year evaluation. But then as we did our own evaluations, we identified some of the PI lines of things that were as susceptible or more susceptible. So we also continue to include those. And again, just to review from 23 at multiple locations and different maturity groups, 2 and 3, we also see significant differences in response from 0 or very, very low to completely susceptible, and over with significant presence of larvae. Well, depending, in the environments we tested, but these were at multiple locations and states, and then reps. So it's 9 reps in this case over 3 field locations, 2 in Nebraska and 1 in Iowa. And so the consistent results showed us there were differential response. There's lines with lower damage and lower area under the severity progress curve. So that's potential resistance. And lines with higher damage in AUSPC. So those are probably our most susceptible. And the differences are highly significant. And we continued with evaluation of lines that were in our breeding program. We didn't have, or Justin didn't have access to a lot of commercial lines along the way, but again, the consistency over years showed that in our breeding material and elite lines, we saw a whole range as well. And so we have really susceptible check, and then skewed toward resistance in our lines. And that may be an artifact of maybe because Gall Midge showed up in Nebraska before we really knew about it, and maybe some of that selection was occurring over time. In 2024, we did a yield test of elite lines, and I'm just showing the results for unhilled plots that were in both Nebraska and Iowa locations. And these are ordered by AUSPC from left to right with the line number on the bottom. And so it wasn't a huge test. We have group 2 and group 3, but we still see, and then the little boxes inside the graph show the yield. So we didn't have a high level of infestation at the sites this year. And you can see didn't really affect yield except maybe that most susceptible one, which the susceptible, what we identified as susceptible in our tests are colored orange in this plot, and the blues are supposed to be the resistant. So there is a little flipping, but we also didn't have really high pressure, as you can see, and yields were from even the worst yield of 67 bushels to almost over 90. So, and then Justin was able to evaluate some commercial lines, and here's a graph showing the result of that. Again, commercial lines do show at at 2 locations in Nebraska, some differential response, and he's showing the top 10 lines at both locations in this graph. So the conclusions are there is a significant potential for resistance in elite soybean germplasm. And that's good news for farmers and companies because you can screen the current elite germplasm phenotypically and genotypically with our genotype data for SGM and make recommendations for SGM infested areas where it's known to be an issue. And at this point, it took us those years to narrow down. We can't work with 700 or more, or even 100, or even 50 accessions or lines on really detailed studies. So now we're at the point where we can, work with yield testing on some lines, do hilling and unhilling, get estimates of loss, test other management practices, maybe with resistance included as a variable. And we're actually crossing now in our Puerto Rico nursery to develop resistant by susceptible crosses and populations to identify or for more detailed studies with entomologists and others who could maybe with controlled environments and colonies and kinds of things, or even in the field, look at a lot more detailed information on a smaller set of material and start to evaluate resistance mechanisms, which could be different since we have so many genes segregating in these populations, and identify potential genes that are involved in those reactions. Thank you.
Thanks, George. I think we will transition to the next presentation with the sake of time, and we'll save the questions for the panel. Let's see, next up is Elliot Nell. He's a research coordinator and he works in the McMecken lab at the University of Nebraska in Lincoln. And he's going to answer all those questions that you guys have been sending in about mowing and tillage. And we hope he gets to that. But I hope at least at some point you will define what you think hilling is. There's been some questions about, what do we describe as hilling with some of the presentations today. So
sure. Yeah. Thank you very much for the introduction. My name is Elliot Nell. I work with Dr. Justin McMecken here at UNL. And I'm just going to be doing a brief overview on on a mowing study that we did, as well as some tillage studies that we did. So just to give you that brief overview, that refresh, if you will, on soybean gall midge ecology. We know that adults are coming from last year's soybean field. This year's usually in a corn-soybean rotation, this year's cornfield, and they're infesting an adjacent soybean field. And what we have often seen over the last few years is if you have dense vegetation between these two fields, or at least adjacent to that soybean field, or this year's soybean field, such as a tree line or bushes, we tend to see greater pressure. And we actually used this for our soybean gall midge survey over the last few years to kind of find and target certain fields, and we've had pretty good success with it. So we developed a mowing study in which we wanted to put this into a data format. So with this, what we did is, as you can see here, we have a grass strip between a corn and soybean field, and we would mow sections of this grass strip throughout the season. We'd keep it mowed throughout the season. We let the other portion grow, as you can see in that photo. And so throughout the season, we'd hopefully get something like this, where we have this very low mowed very low mowed vegetation compared to this dense vegetation. And what we did with these soybean plots is the soybean plots adjacent to those mowed or unmowed sections, we would take infestation frequency. So look at the proportion of infested plants. We'd go through and count the number of larvae, and then we would also take injury scores throughout the season to determine, dead and dying plants in those plots. And then we eventually did take yield. And this is just going to be a brief summarized table of what we've seen over the last 3 years. And this was in conjunction with Minnesota. So in 2022 and 23, we actually did see a decrease in larval abundance, which is good to see in the soybean that was adjacent to that mowed plot versus that unmowed portion. And then if we were to look at that proportion of infested plants, we didn't see any differences there. So no differences there. But if we were to look at plant injury, in 23 and 24 in Nebraska, we did see a decrease in injury, which was exciting to see because that kind of lines up with that larval abundance data that we took. And also keep in mind, across all these years, unfortunately, we had low soybean gall midge pressure. So this wasn't in a very high pressure situation like we wanted. So it's still relatively low pressure compared to a high pressure field. And finally, if we were to look at yield, we didn't see any differences until last year, actually. And last year was really low for soybean gall midge pressure. And that actually resulted in about a 4 bushel increase from what we've seen. So a very minimal increase, but an increase nonetheless. And that was just a brief overview of what we've seen in mowing. I kind of went through that pretty quickly. But moving on, excuse me. Why might we be seeing these differences? So I know we've mentioned that gall midge really likes moisture. And one of our theories is that potentially this dense vegetation that's sitting by the soybean field tends to hold on to moisture. And maybe those adults moving to that adjacent soybean field, maybe it's using that as a reservoir for moisture and increasing its survivability capabilities. Another thing to consider is corn. So as you can see here, the corn in this photo is much denser than our grass strip. So it can hold on to moisture a lot longer. And it also kind of brings into question corn planting date. I know a lot of us don't want to think about corn planting dates, but the earlier you plant your corn, the sooner it's going to canopy. And that canopy, it can maybe hold on to water more. You're going to have more shade. We don't know how that impacts gall midge versus a later corn planting date. So there's something to consider, at least in that regard, which hopefully maybe we'll be looking at in the future, but not entirely sure. So moving on to some tillage comparisons that we did, and this was actually in the same area. We compared a no-till situation versus a disked situation where we disked before planting. And then in our third treatment, what we did is we disked before planting, and we actually hilled later in the season. So we hilled actually after the overwintering generation had finished emergence. And we really did that because we wanted to isolate the damage and injury from that overwintering generation. And just to answer Erin's question, or the question that she brought up, yeah, so hilling, basically what we did is we came in later in the season and we piled up soil to cover that area of the soybean plant to basically remove the area that gall midge can infest. So hopefully it kind of keeps it out. And this photo actually on the top right kind of shows how effective hilling is at the beginning of the season. So if we hill at like V2, V3. And so moving on with this, we went through and we did the same data collections. We took a proportion of infested plants, larval abundance, injury, as well as yield. And I actually lumped these two treatments together, the dist and then the dist and hill, because they responded very similarly versus the no-till situation. So with the tillage and the hilling, we saw an 11 to 26% decrease in infested plants, so less proportion of infested plants. Larval abundance was kind of all over the place. We saw anywhere from no change to a 58% decrease in larval abundance. And then for plant injury, it was kind of the same. We either saw no change in plant injury or up to a 75% decrease in plant injury. As far as yield, 2023, we didn't see any differences. 2024, however, in those plots that we dissed and then hilled later on in the season, we did see a 14 to 17 bushel acre increase. Now, don't know if that was due to gall midge, because keep in mind it was a very low soybean gall midge pressure year. So to kind of take that yield data with a grain of salt there. So why might we be seeing these differences? Well, there's a few theories. When we disc that ground is a lot softer. So that no-till situation, when you go to plant, that surface of the soil is not going to change very much. versus the disc portion, you're going to have kind of that compression from the press wheel, the planter. And that kind of creates kind of an impression in the soil. And it's possible throughout the season, or at least earlier in the season, you can have the soil kind of pile up and potentially hide that or reduce the availability of that plant to gall midge. So that's a potential that it may be possible that's what's happening. Another thing is we may just be making the microclimate kind of not favorable for soybean gall midge. Either that soil is darker, it's absorbing more warmth or more light, so it's becoming warmer. Maybe that's unfavorable. We're kind of changing the residue there. We're kind of incorporating that into the soil versus a no-till situation. So yeah, there's a lot of questions that this kind of brought up. Why are we seeing these differences? And again, low pressure. We don't know what this would do in a high pressure situation. So potentially in the future, we may be looking at that. But that was very brief. And if you guys have any questions, or if we have any time, I'd gladly answer them if I can. And thank you.
Thanks, Elliot. There sure were a lot of questions. I'll probably just keep them to the end, just to make sure that we do a rapid fire for you. So
Sure.
Thank you for that.
Yeah.
All right, our last presentation, we started with Justin. We're going to end with Justin. Lots of just questions about planting depth. And so he's going to share a short update, and then we'll wrap up the webinar with another panel discussion. So Justin, I can see, there you go. Looks good. Go ahead.
Hey, second round. Now I've done it correctly. Okay. I'm going to cover a study that was done by a research scholar in our lab this past summer, and it feeds off of what Elliot was discussing, which was, we were seeing these differences in tilled areas of the current year soybean field. And Elliot kind of posed the question at the last, which was, is this greater planting depth and soil moving in and covering up those fissures, or what's going on? And so he decided to pursue this from a different avenue. And this is Julia Poison's work from this past summer. So to start off, what was the project? It was essentially to plant soybean at different depths along field borders in a randomized complete block pattern in the field. And we started at a 1/2 inch and worked our way down to 2.5 inches. And this was just an initial shot to try and understand what role this might play with soybean gall midge. And certainly, as you could guess, there's complications with planting depth, and we'll discuss some of those. The first one was plant stand. We came in and evaluated plant stand, between about 25 to 30 days after, everything went in, and not surprisingly, we saw some variation in plant stand with depth, but it was that middle 1.5 inch where we got about 105,000 plants. That was greater, significantly greater than planting really shallow or really deep. In this study, this is a combination of 5 sites brought together for this, what you're seeing. So we had 5 different locations for this study. Obviously we were interested in fissure exposure. So, really from soil surface to the cotyledonary node, how much of that fissure was exposed? We couldn't hide it, obviously, with the depths we had. But we did significantly reduce the amount of fissure exposure when we got to that 2.5 inch depth level. So we went from about 0.81 inches showing to 0.78, not a huge shift with either one of those, to 0.66, but still quite a bit of fissure area showing. And as you could guess, soybean variety would have an impact on this as well. Getting into the larval infestation portion of this, we did 2 sets of counts. This is the first one in late June. We pulled plants from the outer rows of the 4 rows of this study that we had. The inner 2 were for yield. And you can see that larval numbers are highest in the shallower plantings. And then at the deepest planting, we have a reduced larval count. Julia took a lot of observation related information from these plots as well. We did see stage differences. So some of this may be attributed to plants that were a little bit behind in development. So just as a cautionary tale to what we're looking at, and we could try and sort that out this coming year. It's just challenging. When we move into early August, you see things flip. Okay, so we still have significance on the deepest planting, but now it's the most infested of the group. And I'll weigh in a little bit with a student's project that was conducted, and actually she just graduated, Pragya Gupta. She looked at the timing of infestation. Once we get past R1, early flowering in soybean, beginning flowering, it's hard to cause impacts in soybean from soybean gall midge, even if we let large quantities in. And so these later shifts may be important, but bigger plants may mean less impact. When we look at plant injury was lowest in the shallowest plantings and higher, but there was no statistical differences in plant injury. These are low injury scores. We had higher injury scores at some locations. Gall midge is challenging to work with. We always seem to put it in the site where it's not as bad for some studies. And then yield, that deeper planting had the biggest impact on yield, so it reduced yield the most. The growers that we talked to at some of these research sites were really interested in going a whole lot deeper, to the point where soybeans would not emerge as a test to determine if we could in fact hide fissures. And so even with these a bit of discouragement in terms of the yield, they want to see if that's something that could potentially be done. I want to leave a couple questions for you, more so than answers on this particular study, which is, the use of an aggressive row cleaner on the planter we use here in Nebraska. They float, much like the photo you see there. But fixing those row cleaners, potentially pushing some soil, getting back to Elliot's conversation about this tilled area. Can we create kind of a concaved area where soybeans can emerge, and then with rainfall potentially close that gap in? So that's one thing we're going to look at. Obviously, growers mentioned planting deeper. This till versus no-till scenario. All of this study was put into no-till situations. And then obviously the varietal differences that might exist with something like this. So that's pretty quick and hopefully to the point. I wanted to leave a minute to prompt you all to help us out a little bit with a survey we have. This is right on the Soybean Gall Midge Alert Network website. So as soon as you log into soybeangallmidge.org, that webpage comes up. Right underneath it, you're going to see this emerging soybean pest survey. And we really need your help to fill out this survey to give us an idea what's challenging you most in your area. It's an attempt to gain an understanding of what's critical to your system. It takes about 15 minutes to fill out. There is a chance you could win a $25 gift card. But we'd really appreciate if you took time. If you scan it with your phone right now, it'll take you to it. But you can also just come back to the website and take that survey as well. So with that, happy to take any questions. I think we're into the question period anyway.
Yeah, thanks, Justin. Could you just remind folks what kind of equipment did you use to hill the soybeans?
Yeah, it's a Hinneker hiller. They're out of Minnesota. There's a couple different series of those. Ours was a 6000 series. Yeah, they're a little bit pricey to get a hold of, and not a traditional piece of cultivation equipment for soybeans. As a lot of people asked questions about how close is this to cultivation. Yeah, it's a form of cultivation, but it's not what we typically think of, especially in soybeans, moving that much soil around to the base of plants in a row cultivation. Obviously for weed control in the past, but you've got to have pretty high speeds to replicate what would be hilling in the studies we're doing.
Thanks. A question for George. When it comes to the fissures that Justin was just talking about, do any of the germplasm that you're looking at maybe have different or more or less prominent fissures, just like the architecture of the plant?
Yeah, good question. We looked at that a couple of years ago because when we first realized that and noticed on the validation population, it looked like a lot of those didn't have fissures when we were growing the seed increase. And so the last couple of years, Justin's smiling, but that was something to evaluate in the plots. And there might be a difference in like the degree of splitting, but we didn't, when we actually looked closely, and by we, I mean just the entomologists, we didn't really see any plants without fissures. So It looks like that's, that's not a thing. And then we accidentally planted too deep one year and had fewer symptoms. And so that's where the planting depth study came from.
Okay,
But yeah, they all have some kind of fissure, as
okay,
you can tell.
got it. Thanks, George. Just a quick question, Justin. Has soybean gall midge been found on yellow and white sweet clover? Yes, okay.
Yeah, Molly Darlington, I think, is on it. Molly has been, she was a postdoc in my lab, is now up in Minnesota. Molly did a lot of work on that, as well as some previous students and people worked during the summer. Both support soybean gall midge. I think if Molly were to chime in, she'd tell us that you'd find them on white sweet clover a lot later in the season than you do yellow. Yellow tends to mature a little bit earlier, but neither one seems to stand out as a better host to look at, like, and they might be even, I was trying to understand from plant taxonomy if they're truly different or not. But either one is worth looking at pretty closely. Look for dark discolorations, much like soybean, but look at knee level and even some grade appearance. That's a good part of the tissue to peel off the stems.
Okay, thank you. A couple questions for Natasha. It's a question was, what the seed applied rate at your 140K unit? Can you just confirm what the imidacloprid was, the rate?
Okay, I think this question would be for the research that we did in the field.
Yes.
So in 2021, we used 0.12 micrograms of active ingredient per seed. And then in 2023 was 0.23.
Okay. And then for your lab study, the LC50 that you were looking at, how does that compare to field rates of imidacloprid?
Yeah, this is a good question. I was trying to do my math and compare the results that we got from the bioassay and the rate from the field. And I saw like the concentration in the bioassay would be a little bit lower than the rate in the field. But is as I mentioned in the presentation, it's important to remember that the seed treatment, like for the bioassay, was the imidacloprid diluted in water and the larvae was in contact, right? So the exposure was higher. And then as in the field, this insecticide will go throughout the plant. So we are still not sure how much of that rate that we apply in the seed will be for the gall midge.
Yes. Okay. Thanks, Natasha. Bob, you talked about a fungal strain that could impact emerald ash borer. Do you know if there's anything like that that has been evaluated for soybean gall midge?
Yeah, so Bob Blanchett is in the plant pathology department here, and he's doing work looking at how effective these different fungal strains are that he's finding in emerald ash borer infested trees, how effective those are at killing emerald ash borer. Those specific strains, none of those have been commercialized as far as I know, but some of those species do occur in commercial formulations, like Bavaria and I think Metarhizium, so that they're like different strains of that same species. Now my mind's wandering. What am I touching on your specific question?
Yeah, no, I think you answered it. As far as I know, some of the like Bavaria and Metarhizium, not necessarily persistent, maybe. So it'd have to be like multiple, like frequent applications from year to year. So I don't know if they persist in the soil, but I think they would be commercially available for things like grubs and things like that. I don't know. Yeah, I think
I guess I'm not sure how much carryover persistence there is going to be from year to year. I'd have to look into that. This is kind of a new world for me working with these fungi. The interesting thing is, sometimes these things can establish within plants, and have some impact that way.
Yeah. Thanks, Bob. This is a question for maybe Justin or Elliot. There was quite a bit of data today and previous webinars that hilling has some impact on suppressing soybean gall midge, but it goes against the trends of some farmers that don't want to use that. So how do you balance that conundrum?
So when we found hilling, I laugh because I've got to tell people the story. When we found hilling as a strategy, I was like, we have solved the issue on soybean gall midge. And for these growers that are really impacted, they can rely on this. And I talked to a few of them, and they're like, nope, not doing that. I mean, that's so far off of my traditional production practice. Narrower row spacings, this is really only for 30-inch rows. And as I gave talks over time, I declined from 20% to 10% to 5% to no one, that it's really just a tool. There is a select few growers out there that are using cultivation, as part of their weed control. And if they got enough speed on that cultivator and they're out there early enough, they might be throwing soil. They'll have to get out of their tractor, walk back and look and see, did I cover up that cotyledonary node to know if they've got something there or not? But I know, especially Butler County, a few field days, there were growers there that were like, I can do this, and I'm having enough weed control issues, I'm going to try it.
Okay. I don't know if this might be a question for George or anybody else, but on some of those germplasm screens where there didn't appear to be as much injury, how do we know if the midges, just aren't doing as well, or maybe they're not utilizing those plants as hosts, like they're just moving to other plots? How do we know?
Yeah, good question. It would be better for the entomologist, but I think I was just sending you guys a note to address that. It's a good chance to talk about where we're going next. And where, what progress we've made narrowing down from 700 to 10. And so now we can look at questions like choice and anti-zoonosis versus some kind of antibiotic reaction in the plant. Does it kill the midget? Does it kill the larvae? And so that's where we are. I don't know what to answer on that right now based on the data we have, but that's kind of what we're leading up to with, we're going ahead and making 20 new populations with 2 really susceptibles and the 10 most resistant. But before we start evaluating hundreds of things from 20 populations, it's something that the researchers can work on with those parent lines that we've used, which come from these last 4 and 5 years of study, and pick which ones, what do you want to investigate about modes of action of resistance? What's it doing to the larvae or the midge, or in preference, or whatever other questions you guys come up with. So that's what I see where we go from here.
Yeah, it's a logical step. Now that Bob in his lab has sort of established a way to have a colony, that gives us a lot of flexibility to do some of those choice tests, longevity, egg production, all those kinds of things that people were asking about biology earlier this morning.
Aaron, following up on that, we were able
Yeah.
to recently secure some funding from the Minnesota Soybean Grower Association, or Research and Promotion Council, to do exactly that, right? Kind of building off of the awesome work that you all have been doing in Nebraska and Iowa and South Dakota with these field screens, and then now hopefully working with that subset of, the most interesting varieties to figure out how they're messing with the insect.
Yeah, really good question. I'm not sure how much white mold is a problem in Nebraska, but maybe more of a problem in Minnesota, maybe northern Iowa. But there was a question about if hilling could lessen the incidence of white mold as well as soybean gall midge. Have you guys done anything interaction between white mold and midges and hilling? It sounds complicated already.
Well, we didn't. By the way, we had a low disease incidence last year. We were scouting for apothecia, those mushrooms that emerge from the sclerotia of white mold, but we didn't find any. I mean, we also were collecting AUSDPC for diseases, but our values were too low. So we didn't find any, I don't say, association between gall midge and white mold in our sites. And also important to say that that was the northeast side that we had, which is the region here in Nebraska that's more likely to have problems with white mold, was lost because of herbicide application. And Dr. Manjo, it's a assistant professor here. He's a phytopathologist. He's also my co-advisor. He had another project in Hull, and he also told me that last year across all his sites, he didn't find much disease pressure. So yeah, we didn't see any relationship between healing and white mold. But it's an interesting question. If we find any, we'll be more than glad to share those findings with you.
Thank you. One follow-up question for the first set of presenters. Just wanted a clarification. Are you saying that soybean gall midge infestations will be less during dry or droughty conditions? I think maybe from what you said, Justin, could you clarify?
Yeah, I love this word, depends.
Mm,
It depends. So I think if adult emergence gets off, and based on what we've seen, rainfall may play a factor in increasing that from a few studies that have been done in Nebraska, and then it gets dry, that might dampen them. How dry we go into the spring, we don't know if we can get dry enough to really impact their emergence. I mean, we've had some dry springs, but we've always seen adult emergence occur. So who knows if we're looking at a wet spring, but if things get dry later, that's where we really get concerned about crop impact from soybean gall midge, because they're in there feeding, and if they're doing well, reducing the amount of available moisture to that plant can increase the mortality and thus the potential yield loss in those fields. I hope that clears it up.
yeah, okay. Yep, I think so. Thanks, Justin.
Hey, Aaron,
Yeah.
just going back to the topic of white mold, another thing to keep in mind is there's a different species of gall midge
Oh, yeah.
that confused us in the earlier years. We were thinking we had soybean gall midge much more widespread, but it turned out to be this completely different species that we're now calling the white mold gall midge that feeds on white mold. It doesn't feed on the plants, it feeds on the white mold that's on the plants, but it's similar sized. The larvae are kind of an orangish color, so something to keep aware of if you're scouting fields, right? If you're seeing fields with white mold infestation and you're seeing larvae specifically associated with that white mold mycelia on those plants, it's very likely this other species and not soybean gall midge. Soybean gall midge is going to typically be at the base of the stems in those lesions that you've saw through the various presentations.
Yeah, good point. I forget about that other little midge. Some questions just generally about performance of insecticides, either as seed treatments in furrow or foliar. Just general thoughts on maybe what's working, what's not working. Maybe interested to hear from a few of you. What do you think, Justin and Bob, Elliot?
Yeah, Aaron, you've had as much probably, shots at this as we had, including, what's been done up in Minnesota. I think the takeaway is it varies. Sometimes we catch these odd studies where things seem to pan out. All of them kind of struggle with yield differences. We had the extreme version of that in Nebraska, where we treated 3 times back to back, 12 to 15 days apart from first adult emergence. We had a significant difference in yield, but it was 6 bushels on what was 20 untreated and 26 treated. So, I mean, those types of studies really produce a lot of caution. All of us are on an upcoming, or at least Aaron, I know you are, and Adam, on a upcoming study with NCSRP to look at different methods of application. We've had some hints with drop nozzles that there might be that, but growers, like a few things we're doing, really dislike drop nozzles. So we're increasing, if we can increase pressure on a sprayer, can we replicate that drop nozzle type? So we see hints. Of efficacy. You feel free to add what you'd like. And Bruce Potter in Minnesota did a lot of work before he retired on it as well.
Yeah. Bob, what do you feel like anything is working better than other things for you?
For insecticides, we're not doing any work yet.
Yeah, insecticides.
In Minnesota, that was Bruce Potter doing that work, and then nothing to add.
Okay. I see. I agree with Justin. It's really variable. I've tried to set up a number of trials, but the pressure hasn't been consistent enough for me to draw a lot of conclusions. So it's hard for me to make recommendations about just even group numbers that work better than others. So that it makes it really hard. I don't know if there's I know some people in the group, Justin and Natasha, maybe have worked with planting date as like a first consideration each year. What is your general recommendation for planting date?
Yeah, this is also a delicate topic because my results doesn't make growers happy. Because what we have seen so far from 2 years of study is when we postpone our planting date for June, that again, I know it's not something that farmers like, we have less pressure of soybean gall midge. What we saw when we have planting date that is mid-April, for example, late April. The pressure, it is high, but when we go to the yield, we have a good yield, right? And this is because about yield, there are other things that impact on the yield. So the amount of light that the soybean can get, a lot of things. But it is important to keep in mind that you can increase your pressure for maybe next year. We saw a window that maybe can be a worse timing of planting date that would be between mid-May, because is when the gall midge start to emerge, and then the plants, they can suffer more from that damage. This is what we have seen so far.
Yeah, great. I totally agree. If soybean gall midge is like your primary concern, to be the first one in the field. I mean, it's a balance between the agronomic benefits and the pest pressure that might happen with the early data planting. Justin, anything to add to that, to what Natasha just said?
Natasha's the expert on that
Okay.
topic. I never have much to add.